Good afternoon, everybody, and thank you for hanging on to listen to this talk, and thank you to Jeremy for inviting me here. So, like Jeremy said, my lab is interested in different aspects of essentially the simple question of, even when you have an immune response to a therapeutic protein, some people develop an immune response, others don't. People respond to different versions of the same therapeutic in different ways vis-a-vis immunogenicity. So essentially, these are the questions that we are trying to address in my lab. And before I go any further, I'd like to emphasize what I'm required to, that these comments of mine are informal communication and they are my own best judgment. None of what I say today is FDA policy, so please ignore everything I say. So we've heard a lot about, in recent years, and as was evident this morning, about immune responses as they relate to CAR-T therapy, etc. But my focus is on therapeutic proteins. And I've taken this excerpt from an article in New York Times, which sort of gives a human face to what it is about immunogenicity. After all, I mean, immunogenicity is not central to the treatment of the disease, except that, you know, some drugs work and don't work. And here in this article, Gina Colata, who's a really good science writer, is telling the story of an individual child, and it sort of brings home why immunogenicity is so critical. And it starts with, she's talking about this boy who has an infection, and he had seemed fine at birth, but soon developed what looked like a bad cold and congestion. When he did not get better, his doctors x-rayed his chest and discovered a very large heart. Then came the diagnostic of Pompe disease in the beginning of his treatments, infusions with an enzyme his body was failing to make. Within a few months, he was learning to sit up and to use his arms. His enlarged heart was shrinking, but his fifth treatment was a disaster. He fell into anaphylactic shock and stopped breathing. The doctors gave him oxygen and epinephrine, and eventually he recovered. 2 weeks later, Maglio received another infusion with the same result. The doctors realized what the problem was. But because Maglio's disease would worsen and kill him if he did not get the drug, his doctors kept hoping he might be able to tolerate repeated infusions. For a year and a half, the treatments continued, and Maglio suffered severe reactions even as his disease was progressing. Soon he could not breathe on his own. At 2 years old, he had heart failure. He was dying. So this is the scenario you come across where you have a really terrible diagnosis. There's suddenly a miracle cure for a child or a grown-up, and then immunogenicity strikes, and you have vanishing options. And very often, patients and their doctors have no idea what has happened. Patients notice only that the drug they take has stopped working. Doctors will switch them to similar drug and hope for the best, but that strategy only works when there are alternatives. And even in the case of drugs like rheumatoid arthritis, at some point you run out of alternatives, and sometimes there are no alternatives. So that's the crucial reason why, you know, immunogenicity becomes so important, specifically, especially in the case of replacement therapies, where without these drugs, you know, people just cannot live. And there is no better, you know, version of better measure of that something has gone really mainstream if, you know, Scientific American puts it up on their cover. And just over a year ago, there was this whole issue just devoted to this so-called war against ourselves, which is basically about immunogenicity. So immunogenicity, you know, as we all know in this audience, compromises the safety and efficacy of protein therapeutics. And it's definitely a priority for regulatory agencies. And besides that, it also adds to the risks and costs associated with drug development, which is not a cheap enterprise to begin with. And specifically because of a lack of non-clinical and preclinical predictive tools, it can be a heavy disincentive to industry to develop medications that address unmet needs particularly for rare diseases where the rewards can be small. And even if a drug is ultimately approved, the human and economic costs of immunogenicity to patients is very considerable. So is the cost to healthcare systems. For example, the model that I work on, which are the hemophilias, which are expensive to treat anyway, costing upwards of $100,000 a year, the cost can very rapidly go to $1 million if somebody develops an immune response. So these are not trivial issues by any measure. So to come to this, what has been, kind of part of the interest in my lab is looking for methods for assessing immunogenicity and how we can use them to understand better what, again, makes one person respond to a drug and not another. So we are essentially concentrating on what people have talked about all day today is the T-cell response to drugs. Okay. Sure. Okay. Okay. And there are numerous methods that a lot of us have used that tell you about different steps in the immune response before you get a T-cell proliferation that can be measured in a lab. And today's talk, I'm going to focus on one such method, the so-called MAPS assay. And the reason that we chose the MAPS assay to answer very specific clinical questions about the treatment of Factor VIII is because of all the assays that we have over here, the MAPS assay gives us some pieces of information that you cannot get from any of the other assays. And that what we are essentially looking for the MAPS from a MAPS assay is an understanding of how a protein has been both processed and presented by the MHC system, which is not a sufficient condition for an immune response, but is definitely a necessary condition for the immune response. And so most of the other assays that we use either tell us about if we want to, for example, determine what a T-cell epitope is. You can have, we saw many talks about using overlapping peptides, and you might use hundreds of peptides, but you might do a really strenuous assay. But at the end of the day, you don't know whether the pre-made peptide that you presented to your immune system will ever be produced under physiological conditions. On the other hand, you can use an intact protein and the assay will work. But when you do that, you don't know whether which of these peptides within the protein is the one that's actually the epitope. And the MAPS assay is the only one which tells you both about the processing of a protein as well as the presentation of a particular peptide. So to take a step back before I talk about the particular project that I'm going to be discussing today, I want to bring up the fact that we have previously seen that this MAPS assay is really useful in terms of an actual clinical problem. So Factor VIIa is a drug product that has been in the market for over a decade, over 2 decades actually, and there are absolutely no reports of anti-Factor VIIa antibodies in hemophilia patients. Now, you have to understand that Factor VII is not used for people who are deficient in Factor VII. Factor VII is a bypass therapy that is given to people with Factor VIII or IX deficiencies who develop anti-drug antibodies. Their protein is no longer able to work. So now you give them excess Factor VII, which is a kind of bypass, which essentially forces the blood to clot. Now, the issue of immunogenicity becomes very, very important in these patients because these patients are not deficient in Factor VII. They have an endogenous Factor VII. That is one reason they don't get they're all resistant to Factor VII. But if you now develop antibodies to Factor VIIa, there is no other alternative treatment for these individuals. You've given them a disease which they did not have previously, and it's very likely to be fatal. So Novo Nordisk started developing a drug which was a variant of Factor VII, which they named Vetreptog alpha. And it seemed to be doing very well till they went into phase 3 trials. And the reason that they made this drug with only 3 mutations is that they wanted to change the kinetics of this protein so that its safety efficacy profile as a drug would be much, much better. So essentially, compared to Factor VIIa, what the mutant version or the variant would do is that it would be much more fast acting, but it would also be cleared very rapidly. So essentially, you would get much superior clotting, especially in situations like emergency rooms, battlefield conditions, etc. But at the same time, because the clearance was so rapid, you would have a much lower risk of thrombosis, which is always the flip side of clotting. So you have coagulation, but you also have thrombosis. So it looked like an almost wonder drug, except that 3 mutations resulted in about 11% of the people in the phase 3 clinical trials developing anti-drug antibodies. And of course, because of the high risk which I just described, Novo Nordisk immediately withdrew this drug from development. And as an aside, Bayer had a similar drug with some other mutations, same problem. They had to withdraw it from clinical development. So now this is a kind of general problem when you bioengineer a drug. So you have an endogenous protein, you bioengineer it, you make some mutations in it, and these will necessarily always be foreign neosequences because they don't exist in nature. I mean, for whatever reason, you've rationally designed this protein and added sequences that don't exist. Now the question is, are the neosequences potential T-cell epitopes? There are lots of engineered drugs which are perfectly fine, nothing happens. Now the question is, before you go into clinical trials, how do you determine if these neosequences are actually T-cell epitopes? And we know that the HLA plays a critical role in the development of an immune response. And the problem then, of course, is that, and that's where the diversity of responses largely comes from, is that you have a very large diversity of HLA variants in the human population. That is the most diverse set of genes in the human genome. So what essentially we are doing in these set of tools that we use is to try and determine which these, in the particular case of bioengineered drugs, these neosequences, how many of these HLA alleles do they interact with, and what are their potential consequences if they do interact. So after Novo Nordisk actually withdrew their drug from development, we worked with them, and we actually looked at a host of, in a post-hoc analysis, we looked at a series of different ex vivo and in vitro in silico techniques to see how well they correlated with the clinical outcomes. So we first looked at, just in silico, do the mutant peptides bind HLA class II molecules with high affinity? And mutant peptides did indeed bind with high affinity to some, but not all HLA variants, which was consistent with some people developing an immune response, where others did not. And then we looked at whether the mutant peptides bind HLA molecules with high affinity in an in vitro assay, and this essentially confirmed the in vitro in silico findings in the set of HLA alleles that we could find commercially available for these tests, for these binding assays. And then we actually did, and this is where it's important in the context of what I'm going to present today, are mutant peptides presented on HLA-II molecules in a MAPS assay? And the answer was yes, we could actually identify the mutant peptides that were generated in a MAPS assay. And finally, we could also show that mutant peptides that bind with high affinity do elicit an T-cell response in a thymidine uptake proliferation assay as well as in an ELISpot assay. And finally, the most important question is, are there any associations with clinical outcomes? And ADA-positive patients all carried at least one HLA allele that binds to mutant peptide with high affinity and was detected in a MAPS assay. But the important thing was, of all these assays that we performed, only the MAPS assay is the one that actually gave us information about, in the terms of the intact protein, whether the intact protein did actually present peptides to the immune system. None of these other assays actually is able to give that information. So we decided to work with CSL Behring and my collaborator in the University of North Carolina to start answering some very specific clinical questions about the use of Factor VIII proteins using just the MAPS assay. So there are numerous Factor VIII products. These are either purified from human plasma, which are called plasma-derived Factor VIII, or generated using recombinant DNA technology, which are in clinical use. So, and recent epidemiological studies and a prospective randomized clinical control trial suggest that the Factor VIII products, that the recombinant Factor VIII products may actually be more immunogenic than the plasma-derived products. The MAPS technology offers an experimental platform for testing hypotheses related to product-specific immunogenicity. For example, the protection of T-cell epitopes by the so-called von Willebrand factor, which is always associated with Factor VIII, has been hypothesized to affect the cellular processing of plasma-derived Factor VIII and not that of recombinant Factor VIII. And these hypotheses can be tested using MAPS assays, which permit the comparison of peptide MHC repertoires when cells are treated with the various therapeutic Factor VIII proteins. So this is what we've been, this is the extent of the assays that we performed. So the source of monocyte-derived dendritic cells were 6 hemophilia A patients and 14 healthy volunteers or donors. And the Factor VIII products that we, as far as they are concerned, there are numerous Factor VIII therapeutics are marketed, but they largely fall into 3 categories. This is except the newest generation of Factor VIII therapeutics, which have either an FC or PEG attached to them. But all the classical Factor VIII proteins are either human plasma-derived Factor VIII proteins, the full-length recombinant protein, or the so-called B domain deleted, where the central region of the Factor VIII, which is not necessary for functional reasons, is deleted to make it just easier to handle and manufacture. This shows a distribution of the HL alleles in all these patients, and whether we found them in a hemophilia A patient or a non-hemophilia A donor, and their distribution in different different human subpopulations. And this is an overview of these 2 groups of individuals or subjects, the number of subjects, the number of unique peptides we found in hemophilia A patients and healthy donors were more or less similar. The total number of peptides were obviously more in healthy donors because the number was much higher. And there was a question about, what length of peptides you normally get. And this is the average peptide length that we found in the 2 groups. And this is the maximum length that we identified and the minimum. So this is the range for each of these groups. And it's, you can see it's largely comparable between these 2. A lot of studies which are done with MHC class II look at the DR subtype rather than the DP. This should be DP and DQ. And this seems to be reasonable based on these data because the vast majority of peptides were found on the MHC class II DR and not on the DP and DQ. And so this is a illustration that I think is important. It looks at ADVAIT, which is a full-length recombinant protein and plasma-derived protein, and we are comparing the location on the Factor VIII sequence where we identified peptides on the different patients. I think this is an important graph for, at least for diseases like hemophilia, because a lot of these studies are done with healthy volunteers, and it's important to determine at some point whether the data you get from healthy volunteers can be easily applicable to the patient population. And here you find that it's almost an exact match between what you find in the patients as well as in the volunteers. So I mean, you don't have to, that means necessarily trouble somebody with a bleeding disorder to donate 500 ml of blood or go in for an apheresis treatment, which is which can be these things can be really challenging for. So I mean, I had several patients who were willing to participate in the study, but they could not, for example, keep their hand out for 4 hours at a time to do apheresis, because I mean, if you're a hemophilia patient, you've been getting bleeding in your joints, etc. So you don't have that much mobility in yourselves, and these are not, again, not trivial problems. So this is a question. So we use this data. This I'm showing it to you for a single patient, and I think I have a graph after this. It replicates for all the other patients. So. And this comes back to, in our earlier studies, and so people do a lot of in silico analysis, or they do peptide MHC binding. And the assumption is that if your peptide binds with high affinity, there is a good chance that it will be presented. And because we had this data from a MAPS assay, we can actually test this and see whether that's the case. So what we did was we looked at 1 million randomly generated peptides. We also looked at 1 million peptides from the human proteome, and we took all the overlapping peptides from Factor VIII and von Willebrand factor, which we were looking at over here, and that's quite a lot of peptides because these are both huge proteins. And what we looked at over here is the in silico peptide MHC affinity. And what we've done here is put them on a percentile rank, because when you look at the absolute binding affinity, it varies depending on the HLA allele that you're looking for. So a high affinity binder for one allele may not be a high affinity. That same value may not be a high affinity value for another allele. So we put all the affinities that you get for a particular HLA allele on a scale of 0 to 100. So now we have normalized all of these. So if you look at any of these large data sets of 1 million peptides, no matter how you generated them, as expected, you would find that their frequency of occurrence will be more or less even across the entire range of affinities. If, on the other hand, now you look at the peptides that you actually identified in the assay and look at their affinities, you find that the curve is shifted dramatically to one part of the graph, showing that you are essentially largely picking up only high affinity peptides in these assays. And this is, yes, I have this graph. So if you look at any of the subjects in this study, you basically find the same kind of pattern, essentially. So that's one question. So the peptides that you identify by a MAPS assay are high affinity, but are all high affinity peptides found on a MAPS assay? And the answer to that question, as you would expect, is no. So this kind of pale gray region that you see over here are all the high affinity peptides that are identified at different positions on the Factor VIII gene. And this is color-coded for different subjects. And there are a lot of high affinity peptides that you find. So essentially, this is telling you that this comes back to the fact that you have there is a lot of redundancy in, if you pick peptides just based on high affinity, you are not going to get necessarily clinically meaningful peptides. Your data set is going to be cluttered with a lot of false negatives. And again, I think that is another really good value in using this assay. So now now we're comparing our products. So you compare the full-length recombinant Factor VIII and the B-domain deleted Factor VIII, and you find that, and this is, again, so the blue shows the peptides we picked up in full-length Factor VIII and beta-domain deleted, and the green graph basically shows the peptides which were just unique to the full-length Factor VIII. And here you see, except for a few peptides that we picked up on either side, most of the peptides which are unique to the full-length protein are the ones in the B domain, which you obviously will not find in the B domain product because they don't exist in the product. So essentially, so basically what this tells us is that though they are made in a different system or made by totally different manufacturers or whatever, the way these two proteins are processed by the dendritic cells, because we are basically giving the two different proteins to the same cells from each individual, you basically find that they are processed in almost identical ways. So as long as they are recombinant proteins, there seems to be very little difference between these two. And now this is the question we had really started out to start addressing, is between the plasma-derived and full-length recombinant proteins, which the clinical studies have. There's a critical mass of data now that has accumulated that from. There may be all sorts of other reasons why you might want to use recombinant proteins, but from the point of view of immunogenicity, the plasma-derived seems to be a much safer product. And here, this is each of these different donors with their two HLA alleles. These are their frequencies on the African, Asian, and Caucasian populations. And across the board, you see that we pick up far fewer peptides from the plasma-derived protein than you pick up from the recombinant protein. And there are no exceptions to this rule. And if you look at the protected regions, that is peptides which are unique to the full-length recombinant factor VIII, across this whole spectrum, you find that they are all over the protein. And the way we did this assay is that, because the argument has been made that the von Willebrand factor is kind of protective to the plasma-derived, that doesn't really make sense in a clinical setting, because factor VIII and von Willebrand factor have such a high affinity for each other that even if the product itself doesn't carry von Willebrand factor, the moment it enters the bloodstream, it is going to bind to von Willebrand factor. So here we made both the recombinant and the plasma-derived free of von Willebrand factor, and then added von Willebrand factor at equal molar ratios to both of them. And so you see that this difference that we are picking up, it comes from the nature of the plasma-derived product, not because of whatever von Willebrand factor is doing. And again, to give a sense of that, so that here I show some, I've mapped some data on the factor VIII gene, which some of the data I've not presented here. But so these pink regions on the factor VIII gene, on the factor VIII protein, are the regions where you find interactions with von Willebrand factor. Now, if you take recombinant factor VIII and just add von Willebrand factor to it, you do protect some sites, and those sites fall squarely where the interactions between factor VIII and von Willebrand factor occur. If you now take the plasma-derived protein, you find the protected regions are not necessarily where the interaction between the von Willebrand factor and factor VIII occur, but are all over the protein. So essentially, this seems to indicate that that whatever it is about the way the protein is made and folded under physiological conditions at a normal rate, at a normal gene expression, is a folding that is either better protected, or it's a more physiologically folded protein, and is processed in a very different way by the immune system compared to a recombinant protein. And there was a question in the morning from someone, I forgot who, about asking in gene therapy that why is it that people have not found, at least to date, even though the patients may seem to be the kind of patients who would develop an immune response to therapy. When they've been given recombinant or they've been given therapeutic proteins, that same patient would be the kind of patient you would expect to develop an immune response. When you make a protein by gene therapy, you're essentially making in vivo, in a human system, a protein which is probably much more closely related to the physiological protein. And this is, again, so whenever you work with trying to make immunogenicity analysis with respect to the human population, you always run into this issue of, how representative are your data with respect to what's out there in the human population. And what we've shown here is the world population, the different HLA alleles. These together collectively represent about 95% of the population, and this is the subpopulation that we have captured in our data set. And you can see here that the relative frequencies for each of them are relatively comparable. And these are just data from, there were 2 previous data sets with 4 and 5 patients each, just looking at it at peptide presentation by presentation of Factor VIII derived peptides in a MAPS assay. And the ones that we found in our assay sort of overlay on those that were found previously, plus we have found many more additional ones, largely because we just have a much larger repertoire of Factor VIII proteins over here. And so our conclusions that almost all peptides identified in the MAPS assay have high affinity for at least one of the MHC variants on that individual. A comparable distribution of peptides with respect to numbers and location are found in hemophilia patients and in healthy donors. Peptides identified by MAPS are enriched for high affinity compared to all overlapping peptides from Factor VIII or 1 Willebrand factor. And significantly higher numbers of peptides are identified on MHC proteins when dendritic cells are exposed to recombinant Factor VIII compared to plasma-derived Factor VIII. And addition of 1 Willebrand, and this is important, factor to recombinant Factor VIII does not make the MHC Associated Proteomics profile comparable to that observed for plasma-derived Factor VIII. And for this particular study, I'd like to acknowledge my clinical collaborators, Nigel Key and his team, and CSL Behring, which participated in this study, particularly in terms of getting us the factors, which are very, very expensive to get, and ProImmune for doing many of these assays. And these are all the people in my lab. And before I leave, I would like to invite all of you and whoever is interested in your groups to a collaborative workshop between FDA and CERC next month on the White Oak campus. And registration is free, but you need to register. And this is the place. It's on October 3rd and 4th. And you're all very welcome. And the basic themes in this workshop will be the advances in understanding the biology of T and B cell responses as it applies to the immunogenicity of therapeutic proteins. And there will be considerable discussion of new analytical techniques and improvements in existing technologies that measure steps in the immune response to therapeutic proteins. And we'll be looking at big data as it relates to immunogenicity from the point of view of repertoires of peptides associated with MHC proteins, as well as in the last couple of years, there's been tremendous progress in getting entire antibody repertoires in patients across longitudinal times, and especially the ability now to get sequencing of light chain and heavy chain parts of an antibody correlated together. And at the same time, there is the ability to get an almost complete representation of the T cell repertoire in individual patients. So these are really, really exciting advances, and this data is the collection of this data is just going to become cheaper, but what you do with that data is going to be more important. And identification of biomarkers for predicting immunogenicity, as well as the application of immunogenicity biomarkers and looking to the future. And of course, there'll be a few talks about the holy grail we all look for, the deimmunizing of protein therapeutics. And we will have some economists talking about the human and economic burden, how immunogenicity affects real people in their day-to-day lives. And thank you so much for listening to me. Thank you very much.
Great, thank you very much, Zuben. And also, I think acknowledgements to Zuben for his tenacity and persistence, because these kind of studies are just so very challenging to get set up, particularly with these hemophilia A patients, that I think the discussion about sourcing, probably going back about 10 years.
Yes, that's true.
It's
Takes a long time.
It's a long time, so you've just got to persevere. Okay, question.
Yeah, so I'll start here. First of all, thank you so much. I love your talk for a
Thank you.
number of multiple reasons, especially because it's a perfect intro to my talk tomorrow. And I can basically cut the first 10 slides of my presentation. So fantastic. Thank you so much.
Thank you.
I missed what kind of prediction did you use for the peptide ranking? It's the IDB, or did you use any other platform?
For what?
For the peptide affinity ranking.
Oh, so we use only net MHC-II.
Oh, net MHC-II.
Yeah,
okay.
because for the simple reason that they have now the largest repertoire of MHC alleles that is available. Plus, if you go back and look at it, they have tested. They have the largest pool of peptide MHC complexes in making putting together the algorithm.
Okay. And very quick second question, the difference in refolding between the natural and the recombinant factor VIII, and the fact that you see differences in the presentation. You talked about possible protection. Are you referring to protection from processing such as peptide or any
processing of cleavage, yeah,
okay,
essentially.
Perfect. Thank you.
Yeah. But on the other hand, we did try. So it's the software that's available for predicting processing is very iffy, and they are available. And when we tried to match them, there was absolutely no correlation. Yeah. Okay.
Thank you very much. Very nice presentation. I will take the different folding proteins to my colleagues that think that any endogenous protein is totally safe and fine. Have both the plasma derived and the recombinant been in the clinic?
For many years.
And do we see a different ADA rate for those molecules too?
And that's why we started this study. That was the motivation for it. That finally, and I mean, for this has been going on for, again, like, these are all things that go on for decades. But just a couple of years back, somebody did a controlled trial and actually showed the differences. Which it's a New England Journal. I could send you the paper. That's.
So then my last quick question. So when you do your MAPS assay, you pulse the dendritic cells, monocyte derived.
I don't do that. I pay money to ProImmune to do it.
So my question would be, do you expect that those results to be the same if you would pulse a different cell type?
Unmet. I think that's a very good point. And I think, as Zuben also said, I'm not so familiar with the comparative other assays we did, but I guess there was correlation between those other assays. Generally speaking, people are using MDC because of the number of cells that you need. But of course, you could work with cells, you could work with macrophages if you wanted to. You certainly could do it with cell types.
Yeah.
I don't know, but generally speaking, I think that's less likely, but we don't know.
Yeah, but what we did do was that the cells from every subject was pulsed with all three, and we did a analysis individually for each individual as well as then the pool data. And there was, of course, each individual based on the HL allele will show you different presentation. But otherwise, the overall pattern essentially remains the same, like, regardless of. Okay, I guess. Thank you. Thank you very much. Thanks a lot.